Hello students, this is Dr. Faizan Mirza. I'm discussing CRISPR-Cas9 system with you. CRISPR-Cas9 system is taken from the bacteria which actually have this system of CRISPR and Cas9 to fight back a viral infection. Gene editing is done using CRISPR-Cas9. CRISPR stands for clustered regulatory interspace short palindromic repeats. This is CRISPR, and Cas9 is a restriction endonuclease. We know that our DNA is a double-stranded DNA, and the DNA is having a template strand across which the mRNA is made. For example, if this is DNA and this is the template strand of the DNA, and I want to just cut this DNA fragment from this DNA molecule altogether. This CRISPR-Cas9 system works very much similar to how we work on the Word document in MS Office, in your laptops or in your tablets or on your phones. Whenever, for example, you want to cut a particular word from a written text, what do? You just go there, you just highlight that particular text, and then you just cut it from the options. And once the word has been cut, we know that the words which are present before and after it, they just fill out the gap, so the gap is not there anymore. What you can do further, you can probably just paste another word here in place of the word that was deleted. Now, this is exactly the analogy that I am trying to portray to you to understand how the CRISPR-Cas9 system works. CRISPR-Cas9 actually is the gene editing tool. CRISPR is a set of genes. It's actually a gene, not a set of genes. This is a set of nucleotides, and this CRISPR codes for a RNA which is called as gRNA. gRNA is guide RNA. The guide RNA is having 20 bases which are complementary to the template strand. So if this is a template strand, this is the test strand, the gene that I want to cut, what I'll do, I need to highlight the gene. How the gene is highlighted, I'll make a guide RNA, and the guide RNA will have 20 complementary bases to the template strand here. The guide RNA will go and bind to this template strand here. Once the guide RNA is attached, we know that this region has been highlighted. Now Cas9, the restriction endonuclease, will go. It will just find out where the guide RNA is attached, because it is having a sequence which is having the active site which is complementary to the shape which is actually present here between the template strand, the guide RNA, and the non-template strand. And this region of the guide RNA, it helps it in aligning a particular in the Cas9 system. Now, this restriction endonuclease will cut this entire sequence. When this entire sequence gets cut, this gap is left in the DNA. Now, once the gap is left in the DNA, the sequence on both sides of the gap will just fuse together, because the phosphodiester bonds can be remade by DNA ligase enzyme, and the DNA can join back. So this is how the gene editing can be done. Now, once gene editing can be done, what's the future of the gene editing tool? Once the gene has been edited, either you can just remove this allele altogether, the gene can just be removed from the cell altogether, or another gene can be placed in its position to replace the gene that was removed. In both cases, the gene editing tool allows you to do anything and everything with gene editing tools. You can just make any kind of cell from any kind of resource, because all you need to do is you need to have a custom made Cas9 which are used for genetic technology. You can alter the genome, and that's how the CRISPR-Cas9 system works. We already have covered genetic profiling, how the VNTR sequences are added to a gel, and how different wells can then run, that can then allow the DNA to run, and we can compare the VNTR sequences for example, paternity testing, or for example, from a crime scene, we compare VNTR sequences. What we need to discuss further is short tandem repeats. Short tandem repeats are STR sequences. STR sequences are short tandem repeats. They have replaced VNTR now. And how they replace VNTRs? Because VNTRs are actually are used though, but STRs have replaced them as well. STRs are microsatellites. What are microsatellites? Microsatellites are the DNA sequences which we use now for genetic profiling, and they have replaced the mini satellites. The mini satellites have now been replaced by them, and why the mini satellites have been replaced? Mini satellites are the VNTR sequences that we used to that were used for this particular process of genetic profiling. STRs are much shorter than mini satellites. They are composed of just 2 to 5 nucleotides which are repeated 10 to 30 times. Most genetic sequences that are just present between the structural genes and are non-coding part of the DNA. Mini satellites and microsatellites are found on autosomes as well as on the sex chromosomes. So we just take these from the cell, from the source. For example, you collect the blood sample from the white blood cell. You can take out these, and you can use the restriction enzymes to cut out these the short tandem repeat sequences. Not just the short attempt repeat sequences, just genetic profiling. Actually, what is genetic profiling? If you just try to understand, and I'm just giving you the details here. Already this is covered in another video here in this chapter. So genetic profiling is basically DNA fingerprinting. We use VNTR or STRs from different homologous pairs of chromosomes. The DNA is extracted from the nucleus and cut using restriction enzymes, the restriction endonuclease. Variable number tandem repeats are the mini satellites, the ones which were used before STRs. Even still, VNTRs can be used. VNTRs are stretched of repeated sequences of nucleotides at a specific position on the chromosomes, and they occupy the same position on the homologous pair. There are 10 to 100 base pairs repeated 5 to 50 times, and these VNTR sequences are also called as mini satellites. So this is just a basic comparison of mini satellites and microsatellites, and both can be used for genetic profiling. When we are carrying out gene therapy, or when we are carrying out gene editing, in fact, we can also use iRNA. iRNA is interference RNA. iRNA can be taken up by the cell, and iRNA then combined with the mRNA to silence the gene. So you can silence the gene using iRNA as well. So apart from CRISPR-Cas9 system, there is another tool, iRNA, which can be used for silencing a gene by making sure that it combines with the mRNA. So when the mRNA is bound to iRNA, it cannot be translated into a polypeptide. When we are going for gene therapy, the gene therapy can be done on a somatic gene therapy or a germ cell gene therapy. Germ cell gene therapy means you are changing the genome of the sperm or the egg. And why are you doing this? Because there is a pre-implantation genetic diagnosis, PGD is being done, and pre-implantation genetic diagnosis means you just, before the implantation is done of the embryo, you just diagnose the gametes with a certain disorder, that whether the gametes or the egg or the sperm are having a normal allele or not, or an allele that you want to be passed on to the next generation or not. If in case, for example, if the sickle cell anemia allele is there, or if the thalassemia allele is there in the egg or in the sperm, and they both are having the same recessive allele, you can just replace that allele with the dominant allele. So in that case, the germ cell gene therapy can be conducted, because pre-implantation genetic diagnosis has been carried out. Now once the normal allele is there, and now you allow the gametes to fuse, in vitro fertilization will occur, and the embryo will be fine. The embryo will not have the recessive alleles from the parents, because if the parents were the carriers of the same recessive trait, there will be 1 in 4 chance that the offspring may have the disease. So if you carry out this germ cell gene therapy, 1 in 4 chance is ruled out altogether, because now the gametes are not having the recessive allele at all, they are having the dominant allele. And even a single dominant allele present in the gamete, in fact, they will be having a single allele only. The dominant allele will then be combined with another dominant allele. So even if you just carry out gene therapy in one of the gametes, either the sperm or the egg, then the offspring will not suffer from the disease, because a single dominant allele will be delivered to the offspring. This brings us to gene therapy for Leber Congenital Amauriosis. Leber Congenital Amauriosis is LCA. It's a rare form of inherited blindness. This is caused by a recessive allele, which is faulty in this case. Vision is poor in the childhood. Complete blindness occurs in adulthood. But we know we have our retinal cells, and the retinal cells keep on regenerating in our eyes, so we keep on seeing the world around us. Retinal cells die at an early age in people who suffer from LCA. There is a gene called RPE65. This is the dominant allele of this gene actually regenerates the visual pigment for rods and the cones. So if you are able to see the world around you, it means that RPE65, the dominant allele, is present in the retina of retinal cells of your eyes. So the dominant allele is there, either both dominant allele or a single dominant allele is there. So RPE65 is there, and you have the normal visual pigment being resynthesized. So people who suffer from LCA, they have a recessive allele of RPE65. When the recessive alleles are there, the dominant allele is absent. It will code for a faulty photoreceptor protein, so the visual pigment will not be resynthesized. What can you do? We can treat Leber Congenital Amauriosis by inserting the dominant allele in using gene therapy. The dominant allele can actually be injected in the form of injections, which are called retinal injections. So we take the DNA, the dominant allele, we just use retinal injection to deliver the gene into the eyes, the retina of people suffering from LCA. We use virus for these as vectors. We insert the DNA into the virus. The virus, which is adeno-associated virus in most cases, it's a non-infectious virus. We inject that virus using injections into the retina of these people. These people are blind. Once they receive the gene of the dominant allele of RPE65 through the viral vector, which is adeno-associated virus, the blindness can be restored. The vision can be restored, or blindness can be restored. I'm sorry for that. The blindness can be treated and vision can be restored. Why LCA treatment is so successful? Because the dominant allele is normal and can be inserted. Recessive will not affect, since the dominant allele has been inserted there. The gene can be delivered precisely into the retina, the light sensitive cell. The immune system is not very active in the eye, so the vector will not be attacked by the immune cells of the patient here. The progress of the treatment can be monitored, that how many more injections are needed for the person to restore his or her vision. The challenges with gene therapy are that genetically modified cells have a shorter lifespan and are replaced by the cells with the faulty allele, because we are not replacing the stem cells. The stem cells are very much there. They will keep on producing the stem cells which will have the faulty allele. So you will have to keep on replacing the dominant allele every time the cell divides. So they need a continuous transfusion or transfection from this kind of virus. Moreover, the allele may not get expressed. Why the allele may not get expressed? Because when we carry out gene therapy, we have to not just add the gene to the vector, we have to add the promoter to the vector as well. And now the promoter will be the region to which the RNA polymerase will bind. And if it's a eukaryotic promoter, then not only RNA polymerase but the transcription factors have to bind as well. So yes, allele may not get expressed in the cells because of the transcription factors not being available or the promoter not being activated. The dominant disorders cannot be treated with gene therapy. Why? Because the dominant disorder, for example, Huntington, it is caused by a dominant allele. So a dominant allele is already there and that dominant allele is abnormal. So you can't treat that with gene therapy, because if you insert the normal recessive allele, the dominant allele will still be there in the cells. The dominant allele will not allow the recessive to show itself in the phenotype. As a result, the dominant allele will still be persisting and the disease cannot be treated. So gene therapy comes with its challenges. So these are the challenges of gene therapy. So genetic modification is not just carried out for treatment of diseases or disorders. We know that genetic modification can be done for herbicide resistant crops or for making insect resistant crop as well. Advances have been made now that we have genetically modified animals as well, and animals which can be of a source of food. For example, you can see this is the salmon, and this is a genetically modified salmon. You can see just compare the size. This the size of the naturally occurring salmon and the size of the genetically modified salmon. They both have a very different size. So what's happening? The genetically modified salmon actually grows very fast, and it attains the full size at a shorter span of time as compared to the naturally occurring or the wild type of this particular non-genetically modified salmon. Why is this so? How the genetically modified organisms are made? We know the genetically modified Atlantic salmon is available in US and Canada and in various places of the world as well. How is it done? Basically, this salmon, the actual fish, actually just grows in its size in the spring and summer. That's it. But if we take the growth hormone regulating gene from another salmon and we add a promoter from another species of the fish, we have added the gene. So what are you having? You are having the gene for the growth hormone. So for example, this is the gene, and this gene is having the growth hormone, and this gene, the gene is actually for the regulating of the growth hormone. This gene regulates the growth hormone. So we have the gene regulating growth hormone here, and this gene is added together with the promoter. So both these things are added together. So this is the promoter here. So we have the promoter and we have the gene. So both these things are added together in the egg of the salmon. Now what do? These are ejected into the fertilized egg. The fertilized egg, will be diploid condition of the Atlantic salmon. Then this will end up having this growth hormone will be produced because of this regulatory gene throughout the year. And because this will be produced throughout the year, it means that now this fish will keep on growing in size, not only in the spring and summer, but all over the year. And that's why it will attain a very large size in a very short span of time, and it grows to this large size just in 18 months as compared to 3 years, which it would take if this gene was not edited in this genetically modified salmon. Now the ethical issues come here, that if we are using genetically modified animal, how can they become a challenge for the naturally occurring animals? So what do? We do not allow these to go in the natural environment, and the females which are used in this genetically modified experiments are the ones who are already sterile. So they coming across the naturally occurring salmon is actually very rare. So in that case, the genetically modified organisms or animals can very well be reared in certain conditions where we reduce their ability to compete with the wild salmon in the natural environment. That's it from my side. Thank you so much.